me?
Yes, yeah.
Okay. My name is Tanya Svitkina. I'm professor at the University of Pennsylvania, and I will be talking about actin cytoskeleton and its role in cells. So this first slide, which I show here, pretty much gives you the topic of my lectures in a nutshell. So actin cytoskeleton, you can see this migrating cell in light blue, in cyan. It plays many important roles in cells and primarily studied for cell motility. And main component of the actin cytoskeleton is actin protein, which is a globular protein which can polymerize into linear filaments. And then this can form different structures like form filaments with the help of additional proteins, and it could be networks which are present at the front of a cell, could be bundles which make it more dominant at the rear of the cell. And all these different actin structure perform different functions, but most of them are related to force generation. So actin cytoskeleton can generate push forces, pulling forces. It can maintain cell shape. It strengthens adhesion and also can move structures organelles within the cell. So now we'll go to detail. And we begin with actin biochemistry, just to know properties of actin and actin filaments. So this is a crystal structure of actin. It is a relatively small globular protein, about 42 kilodalton. It can be viewed as a little bit flattened globules. And crystal structure shows that it consists of 4 subdomains, which name subdomain 1, 2, 3, and 4. So in the presence of physiological salt conditions, this monomer globular actin, in fact, it is frequently referred to as G-actin, which stands for globular actin. So this globular actin can polymerize into filaments, which are called ciprolactin or F-actin. And this actin filament represents double helical structure of actin monomers. And this electron micrograph shows that you can, in fact, see how 2 protofilaments within actin filament wind around each other, forming this double helical structure. So now I want to talk about a little bit about how G-actin polymerizes into F-actin, how actin monomers polymerize into filaments. And we will start with the simplest reaction, when we have pre-existing filament which interacts with the monomer, and then this monomer has some probability to bind filament end. And the rate with which it occurs is governed by association rate constant, or K-on. However, after this monomer is attached, it can dissociate with some probability, and this depends on the dissociation rate constant, or K-off. Another possibility that this monomer will stay bound, and another monomer will attach elongated filament. And this reaction is pretty much identical to this reaction, because even though filament is longer, the chemistry of end is the same. So K-on and K-off are the same, and balance between these 2 reactions, association and dissociation, will define whether the filament elongates or shortens. And we can write this reaction pretty much in it's a very simple reaction. So if we want to determine with what rate actin filaments grow, so it would be amount of polymer formed per unit of time, and this rate would be proportional to number of ends which you put into the reaction, the more ends we have, the faster it will grow, the polymer will be accumulated. It also will be proportional concentration of monomers, so G-actin. The more monomers we add, with higher probability they will bind the existing end. So this term is term for elongation. And this term is for dissociation, which doesn't depend on monomer concentration, just depends on concentration of ends. The more ends we have, the more subunits will dissociate, and it will be again controlled by a dissociation constant K off. So now if we do experiment in which we actually measure how much, we put it another slide, so measure how much actin samples per unit time, and this can be done experimentally, so you can put certain number of pre-existing filaments into reaction, certain amount of monomers, and monitor how much filamentous actin appears in the tube. And what we can see that if we do so, the initially amount of polymer gained over time increases linearly with time. But then it slows down and eventually comes to the steady state. This is an amount of polymer which is added is equivalent to the amount of polymer which is dissociated. So, and this can be seen pretty much from this equation. So when we monitor this amount of polymer, which is on y-axis, versus time, which is on x-axis, we see that it will be proportional to ends, but we don't change number of ends. This is as much as we added at the beginning. Amount of GX initially doesn't change much. If we add it a lot, then even though filament polymerize, it changes the concentration of monomers, does not affect reaction too much, and that's why it looks linear, as if ends, G-actin, and KO are constant, and only time changes. However, as we consume monomers, the amount of polymer per unit time decreases, the rate decreases, and eventually it comes to zero. And to determine at which concentration this reaction comes to steady state, we can figure it out just from this equation. So if rate is zero, then this term is equal to this term, which is written here. Right? And from here we can determine that concentration of actin at which system comes to the steady state is equal of K off over K on. And this is what is known in biochemistry as dissociation constant. And in actin biochemistry, it is frequently referred to as critical concentration. Right? So critical concentration is concentration of actin monomers at which elongation and shortening occur at equal rates. So total amount of polymer remains constant. And now let's see other implications of this critical concentration. So in this graph, pretty much shows rate of elongation. Depending on actin concentration. Let me go back to the previous slide for the moment to make one more point. So this slope of leading part of graph really depends on how much G-actin added initially. Right? So if we added very little actin, it would be still linear but slower. If we add much actin, it would be linear but much faster. So in what we do in the next, in this graph, we just begin the reaction with different monomer concentration and monitor at which rate filaments elongate during linear phase. And again, looking at this equation, we can see that when rate is zero, the big zero, which is here, it happens at critical concentration. Where did I put it? Okay, it is here. So the graph intersects x-axis at critical concentration. Now, if we have concentration of monomer 0, then rearranging this equation, we can see that rate would be equal to negative K off. So, and this is how we get this graph dependence of rate of polymerization on G-actin concentration. Okay, so now let's look. So this critical concentration rate is 0. If we have a monomer concentration above critical concentration, we have rate of polymerization positive. It means the filaments elongate. If we have it lower than critical concentration, then rate negative, which means that filaments shorten. So this is a significance of critical concentration. Above critical concentration of monomers, filaments can polymerize. Below, they would shrink or not polymerize if it was no pre-existing filaments. So in this more complicated situation, so now let's start our reaction not from pre-existing monomers, but not from pre-existing filaments, but from monomers. And again, we just do experiment, we monitor how much filament, actin filaments are created over time. And what we see that if we begin with monomers, early rate is very low. The gain of polymers is very slow. Then it takes off and comes to this linear stage we talked earlier and eventually to the steady state. And this slow initial phase is called nucleation, or lag phase, as shown here. So why is it so slow? Again, let's look at our diagram. So if we begin with filaments, then if we look at individual monomers within filament, we see that this one interacts with 4 different subunits. This one interacts with 3 different subunits. And even the most terminal one interacts with 2 subunits. Which the more interaction each monomer has, the more stable it is associated with the polymer. Now, if we try to begin reaction just from monomers, the very first oligomer, which is dimer, would have only one interaction surfaces between monomer, and this interaction is very weak. So there is high probability that this dimer would dissociate back to monomers. That's why K off for this reaction is very high and well exceeds K on. So it's, even if dimers are formed, they're more likely disassemble than gain a third subunit. Yet there is some probability that this dimer encounters third subunit and they make a trimer. So now this complex is a little more stable because each monomer has 2 bonds. And this, that's why dissociation constant is not as great as for a dimer, but still much higher for this monomer filament because it contacts 3 monomers. Maybe equivalent to this one, which contacts 2 monomers. So this trimer is a little bit more stable, or maybe significantly more stable, so it can gain 4th bond, and now this tetramer is pretty much equivalent to the end of filament, and this is the stage where reaction gains this linear rate because it is equivalent of having pre-existing filament which would bind monomers. Okay? And I just want to acknowledge scientists who really figured out this polymerization mechanism. It's Japanese scientist Fumio Osawa, who condensation polarization or polymerization S cooperative condensation. So pretty much he tested many different salt conditions where monomers convert to polymers. I think I forgot to mention that some salts are needed for G-actin to assemble into filament. So this is how experiments are done, that you add salt and actin begins to polymerize. So Fumio Osawa also found in his experiments that there is a lag phase polymerization, and he figured out that this is because critical concentration exists. So he presented it in a slightly different form than discussed, but it is also very useful. So here again, this is total amount of actin from which we begin reaction, and then we can monitor amount of polymer which changes over time, and amount of monomer changes over time. So if we look at the amount of polymer, then monomer concentration below critical concentration, we don't get any gain of polymer. It all remains monomeric. But above critical concentration, it begins to polymerize, and the more actin we have in the solution, the more polymer will be formed at the end. However, if we look at concentration of G-actin, then if we look at the range below critical concentration, then the amount of polymeric actin would be as much as we added. That's why this linear curve at 45 degrees. But as soon as we exceed critical concentration, all actin which is above concentration will be converted to filaments, and amount of monomeric actin will remain at critical concentration. So pretty much, if you have a mixture of polymer and monomer, so actin filaments, presence of monomers, the concentration of monomers will always be critical concentration. If you increase it, all extra actin would polymerize. Okay? So now, what I describe you is a bit simplified because I treated both ends of actin filaments as if they were identical, but they are not identical. Because if we look at, again, actin monomer, it is asymmetric molecules. It has this cleft at one end and no cleft at another end, and overall these are just different amino acids all over the surface. So when monomers polymerize a filament, they are all oriented the same way. So if we have, like at one end, subdomains 3 and 4 exposed, the other end would have subdomains 1 and 2 exposed. This is what makes this filament asymmetric. The chemistry of one end is different from the chemistry of the other end. And this is what is called filament polarity. Ends are unequal. And this polarity can be seen by electron microscopy. And the trick to see this polarity is to use domain of actin, which is not a protein moving along actin filaments. So this motor domain binds to the actin filament at some angle. So if you saturate actin filament so that each subunit binds one motor domain, it creates a chevron shape or arrowhead shape, which indicates that two ends are different. And because of this morphology, these ends go bar bend and pointed end for this arrowhead shape. So this is structural of actin. But in fact, there is also polarity. And this can be seen using this arrowhead, margin decorated actin filaments with arrowhead pattern. So now if you use this actin filament as pre-existing filament and add monomers, look at these filaments after some defined period of time, then turned out that segment which elongated after addition of monomers at the barband is much longer than this short fragment which polymerized under the pointed end. And this indicates that association rate constant for barband is greater than association rate constant for pointed ends. So these ends are kinetically different. One polymerizes monomers faster, another slower. And remember, this is at the same concentration of monomers, so we talk only about rate constant, which is different. There's yet another difference in monomers, which is chemical. What I didn't bring your attention specifically earlier, but want to bring it now, is that actin monomer is ATP-binding protein. Moreover, it is ATPase, so it can hydrolyze this nucleotide and convert it to ADP. And conformation of actin monomer in the presence, which is bound to ATP versus ADP, is different. In particular, you can look at this loop, which exists as a loop in ATP-bound state, but converts into alpha helix when monomer is bound to ADP. So what is the role of this bound nucleotide? It turned out that it also changes rate constant. Specifically, ATP-bound actin has much greater affinity for barband, or I should say association rate constant, has much greater association rate constant for the barband compared to the pointed end. So then another happens. When ATP-bound subunit binds to the barband, this binding triggers hydrolysis of ATP. So now actin really converts into ATPase. It can now break down this bond, and now actin is bound to ADP and inorganic phosphate. However, when it is bound to ADP and PI, inorganic phosphate, biochemically it is not different from ATP. So this hydrolysis doesn't have too much significance for our reaction. However, when phosphate is released and actin converts to ADP bound state, then the conformational change I talked to you about earlier, this is when it occurs. And this is where actin becomes biochemically different. And because you have more actin added here to the barband, and less actin added to the pointed end, so pretty much over time, this end is mostly enriched in ADP-PI actin, and pointed end mostly has ADP actin. So now we see that two ends of actin filaments are also biochemically different. It is, strictly speaking, different molecule than this one. So they have good right to react differently with different kinetics. And all these constants and association constant, dissociation constant, and critical concentrations, they were calculated for ATP-bound actin, ADP-bound actin, for barband, and for the pointed end, and it may be like too many numbers to remember, but I just want to use it to illustrate the consequences of this differences. So we mostly consider ATP-bound actin as a monomer, because this is physiologically relevant form of actin. And you can see that 12, it means K-on constant, which is 12 for barband, and 1.3, almost tenfold lower for the pointed end, which means that ATP actin would assemble on our band much faster, 10-fold faster. Now, if we look at dissociation rate constant, we see that also for bar band it is greater than for the pointed end, but not 10-fold, not that much. That's why when we calculate KD, which is critical concentration, we see the bar band critical concentration is about 5-fold lower than for the bar band. So what is the significance of that? And to figure it out, let's build graphs, which are called flux curves, which is rate of polymerization versus monomer concentration. So as we talked before, let me put it first, curve for the bar band. As we talked before, this curve should intersect x-axis at the critical concentration. And the critical concentration for the bar band is 0.12, which is here. This curve should intersect y-x at negative k off, which is 1.4 for the bar band. So this red curve shows flux curve to the bar band. And similarly, if we put curve for the pointed, it intersects x-axis at critical concentration. For the pointed end at negative k off, it intersects y-axis, and we have. So then what we can learn from this graph? First, if we look at concentrations above 10 and also positive for the bar band, also it's slow, bar band much higher but positive, which means that both ends will grow concentrations above 0.6 micromolar. If we look now between 0 and 0.12, both rates are negative, so both ends will shrink, again at different rates depending on concentration, but they both depolymerize. But the remaining range between 0.12 and this is where things are interesting. Here, bar bands grow because it is positive, and pointed ends shrink because rate is negative. And depending on which concentration you take on this axis, like if you are doing a reaction, say, at this concentration, you will see that pointed ends shrink very slowly and bar bands grow very fast. Although they behave differently, you will get net gain of polymer. In contrast, if you are here, you will have relatively fast shortening of the pointed end and relatively slow growth of the bar band. So on average, you will lose polymer and eventually all ceramides will depolymerize. But you also can find a range where rate of depolymerization is equal to rate of polymerization. And if all these phenomena between these two critical concentrations is called treadmilling, which means one end grows, another end shrinks, this individual concentration at which both rates are equal is called steady-state treadmilling. So what happens at this particular concentration is that subunits constantly exchange. The monomers are added to one end and removed from another end. Filament maintains the same length, but it is not the same over time. concept was developed theoretically by Albrecht Gardner, who just based on their knowledge that ATP is getting hydrolyzed and therefore ends up chemically different with different rate constant, he figured out that treadmilling should exist. It should be stayed when one end grows and another shrinks. And why I spend this time on this treadmilling process is because it is used by cells. It's used by cells to generate motility. However, if you look how fast it treadmills in our test tube, it is so slow that cells will not ever move at any reasonable rate. In cells, in fact, treadmilling process occurs much faster. And how? Because there's a bunch of proteins called Actin-binding proteins, there are a huge amount of them, and they control every aspect of actin behavior. There are proteins which regulate actin dynamics, this process which we just discussed. So some proteins sequester monomers and prevent them from polymerization. Other proteins would help to overcome the slow rate of nucleation. Some proteins speed up elongation. Others stabilize proteins, stabilize filaments against depolymerization. These helping proteins can bind to filament ends and prevent their elongation or shortening. And there are others which counteract this escaping action. There are proteins which cut filaments into pieces or severing, and those which enhance depolymerization. And there are other proteins which also will talk on the way, which bind filaments to each other, to the membrane, in different modes, so that you can form different structures. And there are also molecular motors which help to move harder along actin filaments. So, and we will discuss roles of many of these proteins, and we will do it by using cell motility as an example. First, because roles of actin are best studied in cell motility, and it would help us to learn general functions of the actin cytoskeleton. But on the way, discussing cell motility will give you other examples where similar mechanisms, similar machinery is used to perform other cellular functions. Okay, so first I probably want to tell you how we currently view how general cell motility occurs. So cell is usually polarized, has front and rear, and when it moves, it protrudes front and retracts rear, and again protrudes front and retracts rear. And there are two different machineries which both consisting of actin filaments, but they perform these two different functions. And this example of actual moving cell, which expresses fluorescently labeled actin, so all what you see white is actin. And as you can see, this is the front of the cell, and it gradually over time approaches the top edge of the frame. This is the rear part of the cell, and you see that it gets retracted and departs from the bottom of the frame. And also this tail gets shortened over time. So in this coordination between protrusion and retraction varies among cells, and I show you two extremes. Like this is fish keratocyte, again some time frames how it moves from left to right. And what you can see that while this cell moves, it doesn't really change shape. And this is very fast moving cell, and its role in fish is to heal wounds if cell damages its skin. So in this cell motility example, protrusion at the front and retraction at the rear are so well coordinated that cell virtually doesn't change shape. And another extreme are neuronal cells. As you know, neuronal cell body is sitting in one layer of the brain, and it extends actually a very long distance away from the cell body to make a synapse to another neuronal muscle. So, and when this axon grows, you pretty much see a lot of protrusion happening in this growth cone, which is also shown here for real neuron, but cell body remains in the same place, which means that retraction of cell body is not really occurring, only protrusion of the front is happening, and as a result, cell elongates. Okay, it's going to move. Okay. Oh, sorry. One should be back. Yes. So these are electron micrographs which show actin organization in the cell. This one shows at the cell front, pretty much equivalent to this part of the cell which I showed you before. And each line here is actin filament. And as you can see, this leading edge of the cell is very rich in actin filaments. But they form two different organization. One are these long finger-like structures which protrude beyond the leading edge of the cell, and they are called filapodia. And they have very long filaments organized into tight bundles. But what is in between is called lamellipodia. It is flat protrusion, and it is filled with less organized actin network, and as I will show you at some point, it is branched actin network. So if we look on the opposite somewhere in the rear where contraction occurs, then you see these bundles of actin filaments where actin cooperate with other proteins, in particular myosin, to cause contraction. So my plan for today, I don't know whether anybody informed you or not, but in fact I'm planning to discuss role of actin cytoskeleton in 3 lectures. So for today I discussed already the kinetics of actin, and pretty much want to discuss how actin polymerization drives protrusion of the leading edge, and today I probably will have time only to discuss filapodia, and for other times I will talk about how lamellipodia protrusion occurs and also how cell contraction occurs at the rear. So, but before I move to the mechanism of filapodia protrusion, I want to discuss just this general idea of how membrane protrusion at the leading edge of the cell occurs. So in short, actin polymerization drives protrusion. So pretty much when polymeric actin converts into filaments, these growing filaments membrane to cause membrane deformation and movement forward. And this concept, in fact, initially caused a lot of resistance. It looked very counterintuitive because the original idea about actin cytoskeleton came from muscle, where muscle was the key protein, and nobody could believe that actin can produce any force without myosin. Yet this concept was established by great scientist Treadmilling, whose scientific approach is quite interesting. He used very unusual organisms, such as this sea cucumber, and he used very unusual cells, which are very specialized cells, which pretty know how to do one thing. And this study I'm going to tell you about, he used sperm cells from this sea cucumber. And the process he studied is called acrosomal reaction, This illustration is from mammalian alpha mouse, but it was discovered using this animal and turned out to be correct for us as well. So what happens during fertilization when sperm meets egg? Sperm has two challenges. One, egg is surrounded by jelly, so this jelly needs to be taken out of the way. And then sperm would meet membrane of the egg and also need to go through. So in order to overcome these problems, sperm has two devices. One is a vacuole of digested enzymes, which opens up and these enzymes are secreted and they digest this egg jelly. And then there is a protrusion of this spike-like structure from the tip of the sperm head, which pokes through the membrane of the egg to allow nucleus to enter egg, fertilize, and get an embryo. So in this C-cucumber sperm, this reaction, which is a acrosomal reaction, is the growth of this acrosomal process, in fact, can be triggered by just adding calcium to the medium, don't need egg. And growth of this acrosomal process occurs at an incredible speed. In fact, this is like a world record in cellular world. 90 micron per 10 seconds. Nothing is happening faster what is given by actin polymerization. Or I will count that it is driven by actin polymerization. So how did Tanya come to the conclusion that this acrosomal process growth is driven by actin polymerization? He just did very careful electron microscopy studies of acrosomal process growth after addition of calcium. So pretty much at the very beginning, when you see a quiescent sperm, you see all its parts. This is flagellum, which moves the sperm. These are mitochondria, which provide energy for the tail. This is nucleus, which obviously has genetic material to transfer to egg. So this is a vacuole which contains these digestive enzymes to degrade egg jelly. And there is another compartment between vacuole and nucleus, which is filled with some gray stuff. So after activation, first enzymes are released, so this vacuole is emptied, and simultaneously a bundle of filaments begins to grow from little stop at the bottom of this additional compartment. And this bundle pushes membrane forward and makes this acrosomal process. And while it is happening, this gray material gets depleted. So Tanya correctly concluded that this is what is used to build this bundle and cause formation of the acrosomal process. And then he proved that this bundle is made of actin filaments. So he did decoration with myosin heads and found this arrowhead pattern. So it proved that this is actin. He also found that barbands are oriented upward over here, which means that this is where growth occurs. So monomers should be added here. And because the reservoir of monomers is at the bottom, then the conclusion is that monomers travel all the way along the process and are added at the tip. And this is in fact a diagram from the original paper. So this is how he presented his model. So as I said, this idea caused a lot of resistance. It was counterintuitive. Why monomers should travel such a long distance to be added there? How polymerization by itself can really do any work? But even after it was accepted, the concern was how general this finding is relative to all other cells which exist in our body. And it was a contributions from other scientists who showed that this is the case for pretty much all cells which are able to move. One piece of data came from Victor Small, who was first really visualized how actin is organized at the leading edge of migrating cells in animal body. So he found what I just showed you before by different electron microscopy technique, that there are bundles of long-acting filaments in filopodia. There is a network of actin filaments in lamellipodia. And also he showed that barb ends of these ends face the membrane, pretty much like in the acrosomal process of scion sperm. And then, of course, it was very difficult to accept an idea that monomers are added between the membrane and the barb end. So, and you really wanted direct proof that this really happens. And this proof was obtained by Yuli Wang. So, Yuli Wang was first to design a technique which we constantly use these days. It means that using fluorescent molecules to incorporate them into cells. So he used actin labeled by fluorescent molecule, injected it into cell, waited when this fluorescent molecule incorporates and mixes up with endogenous actin. So you can see this leading edge of the cell with filopodia, which is bright linear structures, and more diffuse fluorescence in lamellipodia in between. Then what he did, he used bright light to bleach little tip over here. So you see, this is before bleaching, and this is dark area appears over here. So actin there, just fluorophore molecules is killed. So, and this allows you to see how this actin, which was white, now became black, how it behaves. So if you watch the same cell over time, that you see that new population of white, bright actin appears at the edge, and it gradually grows, and the dark zone displaces back. Which means that this is the area where new actin is incorporated into the old actin, which pretty much says that barb ends, which are located at the membrane at the leading edge, they incorporate new actin, they polymerize and push the membrane. So these days it is done by more beautiful, more convincing techniques and more advanced. And just to illustrate this, I want to show you this experiment published in this paper a few years ago from Clemens Rotner's lab. So they in fact expressed two actin forms in the cell. One is labeled with red fluorophore, and it is incorporated everywhere. You can see all actin structures in red. But simultaneously, they injected actin monomers which are labeled with green fluorophore. But this green fluorophore is not visible until it is activated by certain wavelengths of light. So then, they activate this photoactivatable green actin in this region, and now they see both O actin and marked subset of actin structures. And now they see that this green actin gets displaced away from the edge, but new red actin incorporates at the front. So it proves the same concept, that actin polymerization actually occurs between the membrane and the barb end, and actin treadmills backward, because while actin is incorporated here, it must be depolymerized here, because overall this zone remains pretty much the same. And by end, you see that virtually all green actin is gone and replaced by red actin again. And this diagram pretty much shows the concept I just showed you by actual experiments. Then you have migrating cells which has this actin-rich zone at the front, which consists of two populations of actin filaments, phylopodia and lamellipodia. They are oriented as barband to the membrane. So if you label these actin filaments by whatever means, by bleaching the zone or by activating different color of actin, and watch over time, you will see that this zone remains pretty much stationary relative to the coordinate of the cell. And you see new actin assembly in front of this zone and actin disassembly behind this zone. And this is treadmilling. This is treadmilling, which we just discussed at the beginning, that barbands which are here, they polymerize ATP actin, actin gets hydrolyzed and phosphate release, and then this ATP actin depolymerizes from the pointed end. If you have any questions on the way, just feel free to ask. So, and now having this general concept, let's see how filopodia protrusion occurs. And this idea, and which proteins help, and this idea actually also began with this acrosomal reaction in sperm characterized by Ludovny. And the question Luciani was wondering, so if this is a reservoir of monomeric actin to be polymerized on the barbands after activation occurs, why this actin doesn't polymerize by itself? It is present here at huge concentration, way above critical concentration. If we apply just the plain biochemistry of actin, which we just learned, this actin should spontaneously polymerize, and this compartment should be filled with actin filaments. Why it doesn't do so? And the reason is because actin is here not as a pure protein, but it is present, so this is actin in whatever reddish color. It is present in a complex with another protein, which is called profilin. So what profilin does, it binds the barbed end cells of the actin molecule. Right? So again, diagrammatically, this is actin, barband bound to profilin. And what happens then, if you have all monomers bound to profilin, they cannot interact with each other. Because for polymerization, this pointed end need to interact with this barband, and it cannot because profilin blocks. There's another consequence of this, that these complexes actually can be added to the barband, but not to the pointed end. So barband surface of monomer block cannot be added here. But it can be added here because pointed end surface is open and barband is open, can polymerize barband. And as soon as this complex is added to barband, profilin dissociates, and again free barband is created. And yet another function of profilin is that it can convert ADP bound actin monomers into ATP bound monomers, so it functions as nucleotide exchange factor. And another protein which I need to introduce to explain you how filopodia works is a protein which is called ADF-cofilin. And this is a very small protein. And what it does, it preferentially binds to the old filament, which consists of ADP-bound subunits. And when it binds this old part of actin filaments, it twists this filament, so filament breaks. So this ADF-cofilin functions as actin separating. And you can observe this in experiment in vitro. So if you have actin filaments which are labeled with fluorescent molecule, and watch over time, they remain pretty much as they were. But if you add cofilin, they all become fragmented into little pieces. And these little pieces now have many more ends to dissociate monomers. So these little pieces can depolymerize much faster. So ADF coefficient functions pretty much as depolymerizing factor, which actually this abbreviation stands for. Actin depolymerizing factor, and coefficient has different origin of the name, but it is pretty much a family of closely related proteins. And now, having just these two additional proteins, we can build a mechanism, machinery, which would accelerate treadmilling in cells. So if we have pre-existing actin filaments, what is added here is actin porphyrin complex, and it cannot be added here, so it would be added preferentially to the part end. Then this peptide is hydrolyzed, phosphate is released, and this is all ADP-bound filament where ADF coefficient binds, separates, and releases monomers, and then porphyrin binds, exchanges ADP for ATP, and it is added again to the barband. So this cycle, with the help of coefficient and porphyrin, can increase rate of treadmilling about 100-fold. This is the process which actually happens in Filopodia. Again, this is a substructure of one of the protrusive structures of the lineage, consisting of long actin filaments bundled, and they exist in many cell types, particularly abundant in neuroscles. So, and this is how Filopodia functions. So they have these long actin filaments with barband to the membrane. So porphyrin actin complexes are added to the barband. Coefficient acts the rear and severs these actin filaments. And these actin filaments treadmill while polymerizing the push membrane forward, and in the rear they are recycled. But this is not it. In fact, there are many more things which help Filopodia to children, and I will show them to you. So one additional protein need is bonding protein acid. So if these filaments act individually, they are too flexible. So this Filopodia will not have sufficient strength to push membrane. So when they are bundled together, it is stiff bundle, it really can overcome membrane resistance and allow Filopodia to grow. And this bundle also needs to be bound to the membrane, because otherwise it would be, membrane will behave by itself and bundled by itself. You need connection, and this is done by proteins which are called ERM family, stands for first letters of members of this protein, azirin, rhodixin, moisin, and these proteins function to link acid to the membrane. There is a couple of very interesting proteins and very important proteins which discuss. One of them is filament. This protein is sitting at the barb end of actin filaments, and it consists of several domains. One important domain has this donut-like shape, and what it does, it actually allows monomers to be added to the barb end and still be associated with barb end by itself. And it does so because this donut-shaped domain consists of two half donuts, which can have connected by flexible linkers. So they pretty much stair-stepping, like here, this monomer is coming, it is bound here, and this half donut goes up. Then it is strictly bound, another one dissociates and allows another subunit added here. Stair-stepping happens over time, and filament elongates, but forming remains bound. So now filament has these flexible domains, which binds porphyrin actin complexes, and the role of it is that now locally, right at the barb end, the concentration of monomers is greatly increased, and this is what increases polymerization of actin filaments. You remember, rate of polymerization proportional to concentration of monomers. Filament locally increases this concentration and speeds up elongation of filaments. So this is an additional mechanism to speed up processes in the cell. There are other domains of filaments which bind membrane and allow us to keep barband near the membrane and do productive work instead of, say, elongating sideways where it would not create any useful force. And there is another family of proteins which has totally different design, absolutely unrelated to filaments, but in fact does the same job. It has regions which bind to the barband. It has regions which bind porphyrin and actin complexes. It has regions which bind membrane, and it acts pretty much the same way. Increase local concentration of porphyrin and actin complexes and speed up actin polymerization. And different cells use these two protein families, either one or another or both, and we don't know yet how they can choose which one to use. Probably there are some advantages for one versus another, but this is what we still need to learn. And finally, there are family of proteins which are called I-bar domain proteins, which can bind membrane and bend it, because they have this curved surface, membrane binding surface. When they bind membrane is curved. And these proteins associate with the membrane in filopodia and help to maintain its cylindrical shape. So that was conceptually simple machinery of actin treadmilling, polymerizing here, depolymerizing here. Now you can see with all these additional proteins, it's really brought to perfection. So it can polymerize fast, it can maintain shape of this filopodia, and perform this function. So I have a few final slides just to illustrate that this filopodial machinery actually functions in several other cell types and systems. And two examples are microvilli and stereocilia. So microvilli is best studied in intestine. So this is a diagram of our intestine, which has all these folds called villi, and their purpose is to increase surface area of intestine to absorb food and nutrients as fast as possible. And if we look at the cellular organization of this villi, they have these monolayer of epithelial cells, and each of the cellular cells has this microvilli, which fill the increased surface which absorbs nutrients. And these microvilli, like filopodia, have bundle of actin filaments. They are cross-linked together to become stiff. Also, they use different cross-linked proteins. This bundle is bound to the membrane. Again, different protein is functioning to bind to the membrane, but the idea is the same. Barbands, which grow here, are associated with a bunch of proteins which regulate polymerization of barbands. Again, they're not quite clear. They're probably different than those in filopodia, but idea is the same. In stereocilia, these are cells, these are appendages of cells which function in ear for hearing. So this is a diagram of ear where there is this cochlea structure which contains, this is a wall of the cochlea which contains these cells which are called hair cells, inner hair cells, and they have these beautiful appendages called stereocilia. So pretty much when stereocilia vibrate, these epithelial cells begins signaling which lands on the neuron this is how we hear sound. And this is a little bit more detail how stereocilia organize their relatives for macrovilli. We have two different types of bundles, but still barband up bundling proteins, proteins which lead bundle to the membrane, and other signaling proteins which allow it to function. Okay, so that's pretty much everything I wanted to tell you today. And maybe next time, when we choose time, I can tell how laminipodia function, which is a little bit more complicated machinery, but also kind of very interesting. If you have any questions, I will be happy to answer.
Yeah, yes. Yeah.
Okay. Yes.
Yes, she will. So I guess we can discuss the time later on. The 2 more lectures she wants to give. Yeah. Okay then. Yeah. Thank you.
Right. Thank you so much. Okay. Thank you.